Actually, now I'm recording. I just want to, first of all, again, I thank you for coming, and thanks to the students. I think, I know they've all worked really hard on their projects, and of course they lost about a month or so of time to actually get to do their research. So it was especially challenging to pull this all together. But based on the morning presentations, I was pretty impressed with what the students were able to do. Of course, also thank you to the mentors for working with the students as always. I know it takes a lot of time, and I really appreciate your dedication. And I think that's all I really have to say. Unless anyone has any questions or comments before we start, we will. Oh, by the way, too, if you do have questions, you're welcome to chime in. Or if you're familiar with Zoom, there is a little hand raise feature. So, and I'll kind of serve as an MC. So if people have questions, I can pass them along to the presenter. So let me stop my screen sharing here. And so our first presenter is Jolie. She's here. You here, Jolie?
Yep, I'm here.
Okay, so whenever you're ready, you can, let me make sure I actually allowed screen sharing. Yep, so you should be ready to go.
Hello, everybody. So my name is Jolie Ma, and thanks for coming. I did a mentorship with Heritage Trust. Oh, shoot, I forgot to make it a slideshow. So I did a mentorship with Heritage Trust, but because my time with them was limited, I decided to make a presentation over Heritage in general, like its history, a little bit about how it works, and I also did an interview with the founder that I want to talk about. So I'm going to start with the definition of trust banking. And while I have the Investopedia definition pulled up here, trust banking is basically the concept of giving someone else the power to manage a person's finances on that person's behalf. So even though trust banking seems like a very modern idea, the history of trust banking extends really far back before most ancient civilizations, including ancient Egypt. But it wasn't until the time of the Crusades that trust banking became popular as a practice. Because when knights were leaving on their quest for glory, they had to appoint others to manage their finances in their absence. And this is really important because appointing a trustee to manage your finances would help prevent the misuse and abuse of those finances. And also, in the event of the client being unable to carry out their own wishes, the trustee was expected to execute those wishes on that client's behalf. And that notion of putting the client first and always acting in the client's interest helped inspire Bon Payne and Mike Harrell to start Heritage Trust. They wanted to provide a valuable service for those who seek an alternative to big banks. And it's amazing that a company with humble origins, fun fact, Mike and Bon used to operate at the UPO coffee shop, and now they've become so large to have clients in all 50 states. And the merger they recently had with Argent Financial Group only increased its size. So Heritage is made of several different departments, and they can be sorted in 3 levels: the core, the first level support, and the second level support. The trust department, which is composed of relationship managers and associate relationship managers, are considered the core because they're the ones who initiate client contact, who meet the new clients and answer any questions that the client may have, and are just overall responsible for maintaining a good relationship with the client. And because client service is the primary focus of Heritage Trust, the trust department is very important. And that's not to say that other departments don't have their value. The investment department, it manages the client's liquid assets like stocks and bonds. The oil and gas department manages the mineral assets like oil and gas, and they're considered the first level support because they manage the finances. And these departments, marketing, operation, HR, are considered the second level of support because they're responsible for making sure everything's in Heritage is running smoothly. So a typical client meeting who wants Heritage to manage both liquid and mineral assets would have a meeting with a relationship or associate relationship manager, a portfolio manager from the investment team, and a representative from oil and gas. And the relationship manager would lead the meeting while the other two representatives answer any questions that the client may have. So a short while ago, I was given the privilege of being able to interview Bond Payne, and I have a list of many things that he's involved in right here. And as you can see, Bond is very involved in the community and generally a very busy man. So I was extremely glad to have the opportunity to speak with him. And this is the list of questions I asked him, which I will go over. So I asked him what he thought set Heritage apart from other trust companies. And he said because Heritage was founded by trust clients, it gave Heritage a different perspective from traditional banks because it's more because of this, Heritage has more of an emphasis on client service, like always putting the client and their needs first. And they help facilitate this by allowing as much local decision-making and local autonomy as possible. Bond said that it was rather difficult for him to recognize talent, like just through a resume or a job interview, because it's really hard to just get to know somebody and on a sheet of paper just for talking to them for an hour. But he said that he looked for life experience and caring for others because, well, experience obviously gives you a better foundation, but also the more experienced employee tend to be more appreciative of Heritage and what it's doing. And the caring for others, I mean, it's always great to be a good person, but also like it helps with Heritage always like putting the client's need first, and that's a really important quality to have. So when I asked Bond what some challenges that are specific to Heritage Trust or trust banking, he said that a lot of them resulted out of the fact that the industry is incredibly competitive. Because of that, they have to constantly increase their value to their clients, manage fee compression, and increase their innovation. And fun fact, Heritage is always trying to be ahead of the curve. And they were the first to use low-cost exchange traded funds like in the whole nation, and now a bunch of other banks are following their model. But they're constantly looking to innovate and try new things. So the main lesson that Bond learned as an entrepreneur and founder of Heritage is that because he has a pioneer personality, he needs to be able to effectively communicate those ideas and visions that he has, because when he didn't take the time to make sure that everyone understood, everyone was on the same page, then problems usually ensue, because if the other teams don't understand what they're trying to do, it's not very easy to do it. And the advice that he gave for young individuals looking to dive into the finance banking world, either as an entrepreneur or employee, he said to, explore, learn as much as you can, like get internships just everywhere. And he didn't put much emphasis on what kind of internship, like what field, but experience in itself is just critical for building a solid foundation. And he also advise getting a good mentor who you can trust to guide you and who you can trust to answer your questions. And he also mentioned that when you're asking questions, it's better to ask them when you're considered new to the company or wherever you're working at, because you get more leeway then. And I have some acknowledgements because I want to thank people. And I want to thank Bond for indulging me in participating in my interview, my mentors Sadia and Robert for guiding me in their advice, Dr. Richards for always driving me to my mentorship, and my family and friends who are always there to love and support me. And I don't think I could have learned so much without all their help, and I want them to know I really appreciate it. Any questions?
Thanks, Julie. Good job. Did someone have a question? I'm curious about one thing. Actually, looks like Dr. Gibbons says, how has this changed how you think about your own personal finances?
It's changed in fact that I didn't realize like so much was involved in the process. Like I actually my parents didn't even give me my own credit card, but I I just didn't realize like there was so much like behind the scenes, and it's given me more of an appreciation of like finance.
I'm curious about one thing, Julie. You may have said this, but what's the difference between a trust company like this and just a generic like investment bank or like a stockbroker?
They tend to be more like catered to the company's overall interests than like just the client. Like Heritage prides itself on always like catering to the client's best interest, which helps because Heritage is also a privately owned company, unlike many like federal trust banks or just other like government involved.
Okay, great. Well, thank you. Oh, one more question from Agula. Does Heritage Trust manage hedge funds by any chance?
Which funds?
Hedge funds.
Um, I didn't see it. I know they manage, besides the mineral and liquid, they also manage like real estate and also, and which is also taken care of by the trust department.
Okay. Great job. Thank you. So next up is Amy. Amy, can you hear me?
Yes, can you hear me?
I can.
Okay, is it clear?
Yes, it sounds good.
Okay. Okay, can you guys see this?
Yep, it looks great.
Okay, cool. Good afternoon. My name is Amy Brooks, and I will be talking about standardization of immunoblotting of Rho60 and autoantigens. I worked with Dr. Kurian, my mentor, in Dr. Schofield's immunology lab at the Oklahoma Medical Research Foundation. So standardization describes a system by which researchers collect and communicate data. When people talk about data collection, they mean they use a standard scientific tool, a device that is actually capable of collecting data. In this case of standardization, we are dealing with a new piece of lab equipment. Laboratory automation has become instrumental to the progress of the life sciences. Industry provides commercial laboratory devices to perform increasingly sophisticated tasks. However, combining equipment from different providers to work in concert is often difficult. Exporting data for further analysis can be frustrating. Therefore, a protocol is necessary to be developed for this data to be able to be analyzed easily and in coherence with machinery that other scientists use. In essence, this type of standardization tries to reduce the influence of any extraneous variable on the test or experimental performance of the participants. If procedures are not standard, this will affect the reliability of and internal validity and result in biased findings. Findings may fit into a database that other scientists can then access and compare their own findings to. I will give some examples of standardization outside of this for comparison. For example, there are standard terms used in chemistry to describe the bonds formed between each element present in the compound. The labeling of these bonds are the same across the board for scientists, so it's like a common language. Another example is in clinical laboratories. For a specific study, in order to compare the results of one group to the results of a second group, we must make sure that each group receives the same opportunities to succeed. In context of assessment, standardization may define how a measuring instrument or procedure is similar to every patient. For example, a psychologist may adopt a structured interview to systematically interview each patient. By using the same procedures, all subjects are evaluated using the same criteria, and confounding variables are minimized. This is the same kind of idea when dealing with laboratory devices so that results obtained are independent of where and when a test was performed and the measurement procedure used. So we need to understand lupus in order to know what we are even standardizing. Lupus, or systematic erythematosus, is a chronic autoimmune disease that causes inflammation in connective tissues, such as cartilage and the lining of blood vessels, which provides strength and flexibility to structures throughout the body. Autoimmune diseases occur when the body's immune system attacks itself. The Lupus Foundation of America estimates that 1.5 million Americans have a form of lupus, and systematic lupus erythematosus forms 70% of total lupus cases. The causes of SLE are unknown, but are believed to be linked to environmental, genetic, and hormonal factors. As you can see in the diagram here, lupus can affect the skin, mouth, nose, lungs, heart, kidneys, blood, abdomen, muscle, and joints. Symptoms vary, but can include fatigue, joint pain, hair loss, abnormal headaches, rashes, and fevers. These can periodically get worse, as in they can flare up, and then improve. The seriousness of SLE can range from mild to life-threatening. While there's no cure for lupus, current treatments focus on controlling symptoms and minimizing flare-ups. This begins with lifestyle modifications, including sun protection and diet. Further disease management includes medications such as anti-inflammatories and steroids. Because of how serious and common the problem is, our lab is looking into autoantigens that may be the key to developing a cure. To understand what autoantigens are, we must first understand antigens and antibodies. Antigens are molecules capable of stimulating an immune response. They are usually proteins found on the surface of a pathogen. They have distinct surface features or epitopes that result in specific responses. When an antigen enters the body, the immune system produces antibodies against it. Antibodies, also known as immunoglobulins, are Y-shaped proteins produced by B cells of the immune system. Each antibody contains a paratope that recognizes a specific epitope of an antigen, which allows them to bind together. The antibodies destroy the antigens, which are then engulfed and digested by macrophages. There are also memory cells, which remember the same pathogen for faster antibody production in the future. To give a better understanding, I'll tell you briefly how they work with vaccines. Vaccines contain a killed or weakened form of a pathogen. Even though the pathogen in the vaccine has been altered so that it won't cause illness, antigens present in the vaccine will stimulate the immune system and make B cells produce antibodies and memory cells, which makes the body recognize future exposure to the disease. Autoantigens are antigens produced in the body that is recognized by the immune system of patients suffering with specific autoimmune diseases. They are usually normal proteins or a complex of proteins and sometimes DNA or RNA. Under normal conditions, these antigens should not be the target of the immune system, but in autoimmune diseases, their associated T cells are not deleted and instead attack. They stimulate autoantibodies, and lupus is caused by such antibodies. So what are Rho60 and LA? Rho60 is an RNA binding protein that binds to misfolded non-coding RNAs and pre-5S rRNA. It may stabilize some of these RNAs and protect them from degradation. Mice that lack the functional gene encoding Rho60 develop an autoimmune syndrome characterized by anti-ribosome and anti-chromatin antibodies, suggesting that Rho60 may have an important role in preventing systematic immune disease. LA is a protein involved in many various RNA metabolism aspects. In the nucleus, LA acts as a RNA polymerase III transcription factor, and in the cytoplasm, LA acts as a translation factor. Lupus patients can have up to 40% of their antibodies against Rho60, and most patients with the antibodies against Rho60 also have antibodies against LA. These autoantibodies are called anti-Rho60 and anti-LA and are found in the blood serum of patients. What we want to know ultimately is where anti-Rho60 and anti-LA originate in the body, but we have to standardize before anything else. So what we are doing is immunoblotting, also known as Western blotting. Traditional Western blotting is shown in the picture here. This is a method of protein analysis that identifies specific proteins among a mixture of proteins in a given sample of cells or tissues. First, protein samples are prepared and the concentrations of each sample are determined by a biochemical assay. Lysis buffer and a blue dye is added to each sample. After heating the sample to denature the proteins, the samples are loaded into agarose gel, which is put into electrode chamber. Buffer is also added to fill the apparatus. The apparatus is connected to a power supply that provides voltage and the gel runs. This is when electrophoresis takes place, which separates different molecules from each other, which allows you to detect the specific protein you want. The next step is basically to transfer the results from the electrophoresis gel to a blotting paper, as you can see in this apparatus. Then a protein-specific antibody is added to the membrane known as primary antibody to visualize the protein of interest. Any unbound antibody is washed away, like literally washed away with water, and secondary antibody is added that recognizes and binds to the primary antibody. The secondary antibody is linked to a reporter enzyme that produces color or light, which allows it to be easily detected. These steps permit a specific protein to be detected from among a mixture of proteins. When this is done, the blotting paper is scanned by a machine to then be uploaded to a computer. The much faster way to get this process done is through the new machinery that the lab has obtained, which is called JESS. Since it is a new piece of equipment, we had to standardize Rho60 and LA autoantigens. To use JESS, samples, antibodies, reagents, and other materials needed are loaded into a microplate, as you can see in the picture on the right. JESS then does everything a traditional Western blot does, but automatically. However, a camera captures a series of images over time and data is transferred to the computer directly. So it only takes around 3 hours to complete, whereas a traditional Western takes 2 days. Images also have a much better quality. Onto the results. So we discovered that very small amounts of antigen are needed for JESS. We used only around 60 nanograms of antigen, of which the machine uses only about 1 nanogram. On a regular Western blot, we use 1,000 to 2,000 times more antigen. For antibodies, we use only 1 microliter instead of 10 with the regular Western blots. The same goes for secondary antibodies. We use both the chemiluminescence and infrared detection methods. The difference is just that for the chemiluminescence method, a chemiluminescent substrate is used. And for the infrared method, a secondary antibody with infrared dye is used. Here are pictures from traditional Western blots. On the left shows anti-Rho60. It shows up very faintly. On the right is anti-LA, this is still a result from traditional Western blot method, but it was slightly modified with a special gel and buffer, so it shows up better. Here we have results obtained from JESS. The image here is not pristine because I was not able to access the file from the lab, so Dr. Korean just sent a picture he took of the screen on his phone. Regardless, it is still much more clear to see than a traditional Western blot. The red lane is a marker lane, which just lets you compare the size of the protein bands. The next 4 bands, or samples 2 to 5, show anti-Rho60 from lupus patients with decreasing concentration. And then the 4 links to the immediate right, samples 6 and 9, show nothing because those are healthy patients as a control group. Samples 11 to 14 show anti-LA from lupus patients, also with decreasing concentration. Then samples 15 to 18 show nothing because they are also healthy patients. In conclusion, we have standardized the procedure, meaning that we now know how much of everything is needed, including the amount of antigens, antibodies, etc. And we will have a standard baseline for future assays to compare to. With deaths, we save both time and money since it takes a much shorter time to complete the procedure and less materials are needed. And since antibodies usually cost a lot, we save money. The next thing on the lab's agenda is to study anti-Ro60 and anti-LA from Sjogren's patients, which is another autoimmune disease, to understand the autoantigens more and to differentiate the two diseases. Thank you to the OSSM Mentorship Program and OMRF for making this a possibility. Special thanks to Dr. Kurian and Dr. Schofield for letting me join the lab, especially Dr. Kurian for mentoring me.
Great job, Amy. Does anyone have any questions? Dr. Awad says, is there a way that you need a specific way to prepare your samples before running them on the JESS machine?
Oh yeah, well, the JESS machine gives you like instructions to tell you like where to add what in which of the, as you can see here, is where you put your samples and everything. So it just gives you a manual to tell you what to put in where, and then you just put it in the machine and it does everything for you.
And Amy, you said that, I mean, it takes less time, but do you have a sense of how much less time? I mean, for
yeah. A JESS Western blot takes like 3 hours, and traditional ones take up to 2 days.
Oh, okay. Wow, that's a big difference.
Yeah.
But and then you said also it would be cheaper, I assume. But
yeah, because you need to use less of everything.
But is that including the? I mean, I'm assuming this would be an expensive piece of equipment. So obviously,
I think like over time it'll pay off, because like antibodies can cost like $200 like for just a tiny bit, and so you could run so many more samples with like less materials in general.
okay, good job. Thank you. So next up is Adarsh Kunal. Are you there, Adarsh?
Yes, I'm here, Dr. Richards.
Okay.
Good afternoon, everyone. For those of you who don't know me, my name is Adarsh Kunal, and I worked with Dr. Awad at the OU College of Pharmacy to study biomarkers in traumatic brain injuries, or TBIs. One thing to note is that my presentation is built off of literature research of various biomarkers for TBI, since I wasn't able to collect all the data due to the ongoing virus outbreak. So what is a traumatic brain injury? A TBI, as defined by the CDC, is a disruption to the normal function of the brain. It can be caused by a bump, jolt, blow, or a penetrative force to the skull. The severity of a TBI ranges from mild, which is characterized by a brief change in mental status or consciousness, to moderate, then to severe, which is characterized by an extended period of amnesia or unconsciousness following the injury. So this diagram here shows the characteristics of the lobes of a healthy brain versus the lobes of a brain with a TBI. It focuses on the differences if a specific lobe was injured by the traumatic brain injury and the complications afterwards in that lobe-specific functions. So the functions of the brain, like memory, movement, sensation, and emotional functioning, are compromised in one form or another, depending on the severity of the TBI. So traumatic brain injuries are a serious public health issue in the United States, and they are a major cause of death and disabilities. Every year, approximately 1.5 million Americans sustain some form of a TBI. In these 1.5 million people, 200,000 people are hospitalized and survive. 50,000 people succumb to their injuries, and around 80,000 to 90,000 people are disabled for life. So if I try to sum it up, there are an estimated total of 5.3 million men, women, and children living with a permanent TBI-related injury in the United States today. Traumatic brain injuries are often referred to as the invisible epidemic because their disabilities that come from cognitive, emotional, sensory, and motor impairments are not as apparent to the public and often permanently alter a person's lifestyle and have deep effects on their social and family relationships. This graph here shows the leading causes of TBI currently. As you can see, falls make up about half of what causes TBI, while motor vehicle accidents and assault makes up significant minorities for causes as well. So what are biomarkers? Biomarkers in general are characteristics that are objectively measured and evaluated as indicators of normal biological processes or normal health, pathological processes or the course of disease, and or responses to a therapeutic intervention or treatment. They're usually qualitative and quantitative measurements that display at a specific time point or disease state. So in my mentorship, I aimed to find biomarkers that could reveal the health of a patient in relation to the course of their injury. The ideal biomarker would be a blood biomarker taken from a blood sample. Blood biomarkers offer objective information about the health, pathology, and response to a treatment of a patient. One advantage they have over biomarkers that are taken from cerebral spinal fluid or CSF is that they can be taken from a simple blood test, which is much easier than using an invasive procedure to acquire the CSF from the spinal cord. Blood biomarkers are also more efficient than CT scans, which require expensive equipment. So the three potential biomarkers I researched were the glial fibrillary acidic protein, or the GFAP, UCHL1, and the tau protein. So GFAP is a protein derived from glial cells, where it is also the most abundant protein. It is a specific biomarker of diseases in the central nervous system. The blood-brain barrier protects the neurons in the brain from the circulating blood outside of the brain. When a TBI occurs, there will be complications in the blood-brain barrier that will allow GFAP, which is normally found in the brain, to come into the circulating blood. This increase in GFAP concentration from a blood test can be used as a good blood biomarker for diagnosing a TBI. So a study in China with 67 TBI patients from a local trauma center took their CSF samples on day 1 to day 5, and then assessed their health outcomes 6 months later. The GFAP was detected and quantified by enzyme-linked immunosorbent assay, or ELISA, and the patient outcome was determined by the Glasgow scale, which measured injury severity. The study showed that there was an increase in GFAP levels from day 1 with patients who had a severe TBI as compared to healthier patients, and were also predictive of poorer neurological outcomes after 6 months. So this shows that GFAP could be potentially used to predict clinical outcomes in a TBI. The ubiquitin C-terminal hydrolase, L1, or UCHL1, is an enzyme in the cytoplasm of most neurons in the brain. In a case control study with 66 patients, ventricular CSF samples for each patient were taken in several hour increments after the TBI. The ELISA again was used to detect the amount of UCHL1 at these times. The injury severity was also again measured by the Glasgow scale after 6 months. This study showed that patients with TBI had increased amounts of UCHL1 in their CSF at each time point compared to patients who had no TBI. These patients who had these elevated levels of UCHL1 also had lower scores on the Glasgow coma scale after 24 hours, which meant they had more post-trauma complications and even death later on. Thus, UCHL1 could be used in determining the severity of TBI in patients. A really important development to note is that in February of 2018, the FDA approved both GFAP and UCHL1 for a blood test called brain trauma indicator. The tau protein is a microtubule stabilizing protein. It is hyperphosphorylated after TBI and is currently being studied as a biomarker for multiple TBIs, which is also known as chronic traumatic encephalopathy, or CTE. So the goal of my project was to determine the changes in GFAP levels in both serum and cerebral spinal fluid samples, or CSF, collected on day 1 and then 8 days later on day 8, following a mild TBI impact in Sprague-Dawley male rats compared to sham rats. So for both the mild TBI and sham rat models, we used a controlled cortical impact method using a TBI impact 1 device. We used stereotaxic surgery to give this TBI impact. We also used these parameters for the mild TBI rats who had both the craniotomy, which is a removal of a part of the skull, and the TBI. The sham rats, on the other hand, only had the craniotomy, and they didn't have any TBI as well. So in panel A, we conducted a Western blot, only on CSF samples from both the mTBI rats and the sham rats. Here we could see that the sham had relatively little GFAP and tau protein compared to the mTBI. Right there. And this was also shown in the graph below, where this dotted line represents 1 and also the amount of GFAP in sham rats. And so the mTBI rats had approximately 4 times the amount of GFAP than the sham rats, and approximately 6 times the amount of tau protein than the sham rats. So we decided to conduct an ELISA on both CSF and serum samples. In the CSF, we could see that the GFAP increased much more for the TBI compared to the sham on day 1, right there, and then normalized on day 8. This was good as it showed us that the TBI impact in the mTBI rats increased their levels of GFAP. However, in the serum sample, there was relatively little changes in the GFAP levels between the mTBI rats and the sham rats. This could have been due to a small number of rats in our sample size who were being tested on, and or the mild severity of the TBI itself. So the next step would be to continue the studies with more animals and more severe injury to prevent this lack of GFAP changes between the mTBI and sham rats. We would also determine the levels of the next potential biomarker, UCHL1, in the CSF and serum following a TBI. I was not able to collect data on the UCHL1. That's why that is the next step, because of the pandemic that happened. I wasn't able to get data on that yet. So for my mentorship, I worked under Dr. Awad at the OU College of Pharmacy, and also students Dion Robinson, who's right here, Clark Mullenbrock, and Matthew Bayer. I wanted to especially thank OSSM, especially Dr. Richards, for giving me this unique opportunity to have a mentorship in high school, and the OU College of Pharmacy and Dr. Watt for allowing me to learn more about the brain and traumatic brain injuries through this research.
Great job, Arash. So Professor Baxter asked, in your readings over TBI, did you come across anything on sports-related TBI in children and teens? And if so, which sports were most frequently related to injuries?
From some articles I read a while back in my research, it talked about most of the time about concussions in football, because that's a really common thing in that sport, and how it's a growing concern for parents as they send their kids to that sport. And I guess like that's the sport I've seen, the main sport I've seen in my research.
Okay. And let's see, Rahul asked, how did the ELISA work in your experiment?
Okay. So here, I'll go back to that slide. So if you're looking, are you asking about, is he asking about the results or from the ELISA? Is that what he's asking specifically?
Yeah, how did the ELISA work?
Oh, okay. So basically, what? Okay, so basically here it shows like there's two graphs, one for CSF, the sample, and one for the serum sample. CSF is cerebrospinal fluid and the serum is from blood. So here in this graph right here, we can see that, so the white is, the white is the sham and the black is the MTBI rats. So there are two different rats. The sham have only the craniotomy and no TBI, while the MTBI have both the craniotomy and the TBI impact. So here in this graph right here, we can see that compared, so the MTBI here compared to the sham increased quite a bit on day 1 and then normalized on day 8. So we could see that the differences were great between the MTBI rats and the sham rats. However, the serum samples weren't that great because the changes between the sham rats and the MTBI rats weren't as prominent, and there was relatively little changes between day 1 and day 8 for the GFAP levels between the two. So that is why we said it might have been because we used only about 2 to 5 rats per group, or it might have been because we used a mild TBI, MTBI, that's what it stands for. And so that's why our next step would be to add more animals with more severe injury, and then also look into the next biomarker, UCH1.
Okay, and then one more question. Rohan asks, are there any demographics as in age groups most prone to TBI?
Yes, actually. So the age groups that are most prone to TBI are adolescents, young adults, and the elderly over the age of 75 years of age.
Okay, great. Good job. We will move on to the next presentation, which is Josh Lee. Josh, are you with us?
Yes, give me a quick second. I'm just getting things ready. Okay. Let me screen share. Okay, can you guys see this?
Can.
Okay. So yeah, hello everybody. Wait, this doesn't look right. Okay, give me a quick second. Okay, I guess we just have to do it
Well,
this way.
actually, if you go down to that little button that has the dots on it right there, and then hide presenter view, and then
Okay, wait.
and then try switching in Zoom to sharing. There you go. Perfect.
Okay. All right. Yeah. Sorry for that, guys. Okay. Yeah. So, hello, everybody. My name is Josh Lee. My mentor is Dr. Garg from OU Aerospace and Mechanical Engineering, and the project that I did was testing the thermal conductivity of polymers. Okay. So, I'm pretty sure you guys have already taken a chemistry class and you guys know what polymer is, but what a polymer is. But this is just like a review of what a polymer is. So polymers are long chains of similar units. They can be made out of either plastic or organic materials. Famous one you guys probably know is graphene. So some key aspects about polymers and why they're so useful is that one, they're very cheap. So when used in industry, right, compared to iron and aluminum and titanium and all sorts of other stuff, producing polymers is very, very cheap considered to making those materials. And polymers aren't as dense as these metal materials, which makes them lightweight and more applicable to aerospace engineering. They also require less energy to make because all you need is a chemical lab and some ingredients to create polymers, unlike metals where you need time to refine them, to mold them into shape. And lastly, polymers are corrosion resistant. So unlike iron, which can easily rust and maybe break down over time, polymers are they require like pretty much no maintenance. Alrighty. So my project was testing over the thermal conductivity of polymers, and a specific polymer that I used was a combination of polyethylene and graphene. So for this project, I used a vacuum chamber was already pre-built by the grad students, and I was going to use that. And also here's an IR camera that I would have used for the project. So the procedure, so everything begins in the chemical lab, although I wasn't able to go into the chemical lab and make stuff because that's not my level. I probably blow stuff up. But yeah, so grad students created samples that I would use. Yeah, here's polyethylene, here is graphene in the picture over here. And then this is what a sample would look like. And then after they made the sample, they would use this machine in the bottom right to stretch it out. I'll explain why stretching is really important to this whole process in a second. But after they'd stretch it, I would cut it out and make an apparatus of it. I don't have a picture of the apparatus, so I had to draw one on Microsoft Paint. Let me see if I can screen share Microsoft Paint. So here's what the apparatus look like. So this is the polymer. The polymer is usually really small. I'll cut it into like a really small rectangle, maybe about 2 centimeters by 5 centimeters long, or big and 2 centimeters by 5 centimeters long and wide. And after I cut the polymer out, I would cut a little middle section here, as you can see, out, and then wrap a thin wire through it, and then apply thermal paste in the middle. So the reason for cutting this hole in the middle was to ensure that the heat would spread evenly throughout the whole polymer. And then the wire was the way to conduct energy throughout the polymer. And since this wire was very small wires have big resistance, which in turn which release a lot of heat. And then the thermal putty was just to help conduct the heat. And lastly, the thermocouples were there I had to attach them in, and they were used to measure the temperature of the polymer. Let me go back to PowerPoint. Okay. So yeah, so about stretching, stretching is really important because when you make the polymer, they don't come in like perfectly aligned. When you initially make the polymer, they come out in like that picture below to the bottom left. They're all scrambled, everything just scrambled out. After you stretch it out, they become more even and allow heat to conduct easier. So an example of like a microscopic close-up. So before the graphene and the polyethylene, you can see are all bunched up together here, and then after it's stretched, it's pretty smooth. Okay, yeah. So for the testing, after I made the apparatus and I attached the thermocouples, I would put it in the vacuum that the grad students created, and then just pretty much heat it up, make sure everything was working. Use the IR camera as a secondary thing to measure the temperature, just in case the thermocouples weren't working. And the reason we used the vacuum chamber was because we had to ensure that no weird particles would go on would be present while we were heating the polymer to make sure our results were correct. Yeah, so applications for polymers can be, so for instance, aircraft. Polymers can be used in aircraft because they're very lightweight. There's no
Hey,
need for
Josh,
maintenance.
you may want to switch back to your PowerPoints.
Oh, wait, did I not? I thought I switched over. Wait,
That's great.
wait, it's good?
Yeah, now it is.
Oh, okay, my bad. Did you guys see these pictures? Yeah, so okay, yeah. So before, yeah, so it's the stretching part. Before is all tangled up, and then after it's more aligned. And then this is the microscopic image of the sample that I used. And then, yeah, so applications, yeah, they can be used for a variety of things. I mean, plastic bags, for example, shampoo bottles. Before, like more industrial means aircrafts, that you can use it for aircrafts. They're extremely lightweight. They don't need maintenance. And also for pipes, because plastics are non-corrosive. And yeah. So wait, before I go into questions, I just wanted to let you guys know that due to the second semester where Dr. Garg had a lot of meetings and COVID-19, I wasn't able to get any results. So yeah, if you guys have any questions, please ask.
All right, thanks, Josh. Good job. Does anyone have any questions or comments? So, Josh, I'm just curious if if you were to follow up on this project, what do you think like the next step would be, or next experiment you would try?
With like, can you elaborate on that question more? Like,
Well, I mean, like if you were able to keep doing this, like if you hadn't had to stop in the middle, what would have been the next thing you would want to do?
oh, okay. So yeah, so the next thing I would have planned to do was actually start testing the polymer because I didn't get the chance to do that. I would have put it in the vacuum chamber and started heating it up and just doing tests and and graphing everything out. Yeah.
Okay. All right, great. Thank you very much. We will keep going. So next on the list is Jenny. Are you there, Jenny?
Hi, Dr. Richards.
Hi there. Oh, wait, sorry, Jenny. Actually, Abdullah had a question for Josh. So he said, how effective is carbon fiber as a polymer?
Carbon. Oh yeah, carbon fiber. Shoot, I actually don't know much about carbon fiber, but carbon fiber. I'm pretty sure. I actually don't know. All I know is that carbon fiber is a really hard material and it can be used for a lot of things. But other than that, there's really, I can't really answer that question.
Okay. All right. So now, Jenny, whenever you're ready, you can take it away.
All right, can you guys see my screen?
You can.
So hi guys, my name is Jenny Nguyen and I'll be presenting today about the 2 different mentorships that I got to do this year, which is why I'm talking about both ends of the spectrum for speech pathology. So I started out my mentorship at the summer program that OSSM provides, IRSP, which is our Investigative Research Scholars Program. It's a 3-week program where we get to meet our mentors beforehand. So I started at iLeap with Jessica Latham, and iLeap is the Interprofessional Language Enrichment and Pre-Kindergarten Program. And that's a picture of me and Cindy. I'm the one in the scrubs. And the cool thing about this mentorship was I got to wear scrubs every day for like 5 weeks because I actually worked with children with Mrs. Latham. We worked with children who have delayed communication development and behavioral challenges. So a lot of the kids there would be on the spectrum, which means they would have autism. And we actually had one kid who came to classes, she was neurotypical. So she was just an everyday kid like you and me. She didn't have autism, she didn't have ADHD. And we had her there as an example for these kids to help learn and be a part of that preschool environment. So a typical day in that classroom, we'd always start off at the smartboard, and the first thing these kids would see is this video I'm about to show you. And if you can't hear the audio, that's okay, because I'm going to explain what they're saying. I'm only going to show like 10 seconds of the video. But this is the exact video that we would use, and that is actually the classroom that we would be in.
Hey, Jenny, you may have to switch it over to your other screen.
So are you still seeing the PowerPoint right now?
Yeah, we're still seeing the PowerPoint.
Okay. How about now?
Yeah, it looks great.
Okay. So we would watch this video with them every single day on the carpet. And as you could see, they said sharing with friends. That's good. Let me see if I can move back. They would say sharing with friends, that's good, and we'd give them a thumbs up, or not listening, that's bad, and we would give them a thumbs down. And this would just help as like a visual and a verbal way of encouraging them to do what is appropriate in the classroom. And it wouldn't just be in this video when they would hear those words. Throughout the entire class period, whenever they did something wonderful like sharing, or if they started running around and screaming, we would tell them that it's good or that's bad, so they'd know when is the appropriate time to do something. And then after smartboard time, we would give them recess. That's just to get them outside, to get some of their energy out, because a lot of these kids were very energetic, but then also some of them were very reserved. So this gave them an opportunity to kind of be with friends and kind of push them out of their comfort zone. And one of our main things the classroom revolved around was having structure and repetition for these kids. So every day they would go through the 6 major class periods. So there would be math, reading, art, sensory, and motor. Motor having gross and fine. In gross motor, we'd get them to move their entire body. We'd get them active. We'd use things like hula hoops and like dinosaur eggs, because every week there'd be a theme just to get them more excited. And then there'd also be fine motor, where they'd use their fingers and they'd get more involved in like little tasks and more fine detailed activities. So the daily goals for the classroom would be to just help them with their manners. Sharing was a big thing, especially we had this pair of brothers who both had ADHD and autism, and it was very difficult for them to share, especially being brothers. But we always encouraged them to just hand their toys over. And if this corner, we had this little car thing that was outside. That was a really big thing that was hard for them all to share, because they were very excited to just waddle around in that. And we'd also emphasize them saying please and thank you, because a lot of the times they'll just grab and reach for things. But a part of manners and social skills is using your words, which is a phrase we often said to them. We'd want them to ask us thank you for something, rather than just yell and point and just using their manners. And we'd always start the day and end the day with saying hi and bye very enthusiastically to them. And it would always be a blessing whenever they said it back. We just wanted to encourage that interaction. And the overall main thing we wanted for them was independence. It's very hard for these kids to not cling to their parents or just hide in the corner of the classroom. We wanted them to be able to get up and go be their own person and do what they wanted to do and express how they wanted to express that. And a way we would cultivate all of these skills would be through every part of the day, like snack time, story time, even in the bathroom. We'd always be teaching them how to use these skills and how to just express themselves. And then outside of IRSP, I could not continue that after the summer because these kids needed constant interaction, and me coming in and out just like once a day or once a week wouldn't be good for them. So I stayed in the speech and hearing center, but I moved to Mrs. Latham's friend, Dr. Nutero. She is also a speech pathologist, but she was more on the research side. So she was working on a case study that focused on the attention and the focus of children who stutter, because it's often a common notion that kids who stutter are less focused, they don't pay attention as well. But she believed that kids who stutter are actually more focused, especially on certain emotions. So one of the tests we used to try and prove that was these two faces right here. We would show them on the screen, and there'd be other faces with different emotions and different colors. And we'd ask the child to pick the emotion, just pick an emotion, and we'd have a camera on them, and we were wanting to see which picture their eyes instantly went to, and often it'd be the more expressive emotion. So in this case, if they saw a sad blue face and a red angry face, we'd see their eyes dart directly to the angry face, and they'd pick that emotion immediately. And some other tests that we'd go through, these kids would come in on a 2-day period. On the first day, we get to know the child. We get to know if they're quiet, if they're timid, or if they're excited to be there. We'd also take tests to measure the severity of their stutter, just to see like if their stutter was too severe and we really couldn't understand what they were saying, we might not include them in the trial, or if they hardly stuttered at all, they might be an outlier. And then on the second day that the kids came in, they'd go through a series of tests, just like the face test. They do an identification test to see how well they can match up things. Like we would show them a neutral picture like this cabinet or a negative picture like the fire right there. And we would ask for them to pick a negative photo in that moment, something like that. And there'd also be positive photos like kids playing outside. But during all of these tests, we'd always have the cameras focused on the children's fingers so that we could see if they misclicked something, we'd see if that was a misclick or if they actually got that wrong. We'd also have it panned on their face so we know where their eyes were going in that moment. And we'd also have it on the computer screen also so we can see if there was an error or if they misclicked something. A lot of my job in the lab was the case study preparation. I'd often troubleshoot tests. I'd do them myself just to see like if things were working, because we were working on iPads a lot of the times. And sometimes when the test asks you to press the space bar when you're ready to move on, it wouldn't move on. So it was my job to just write that down so Dr. Nutero could go and fix that. I would also organize the data. We had a couple of trials before the pandemic, so I was able to watch through the videos, record what the child answered for every session and the time for all of those answers, and just organize that so it'd be easy to analyze later. Another big part of my job there was transcribing the trials. Because these kids stutter, it's often hard to hear what they're saying, and speech pathologists actually have a sort of code whenever transcribing. So whenever they'd stutter, I'd write down a certain word and this how many times they would stutter and what word that was. And I would also just write down what the tester is saying so that it's easy to read while you're watching the video. So a big part of both the clinical and research side that I got to experience was just helping these kids that have communication disorders and delayed development, whether it be in the home, in the classroom, in everyday discussion, and just in these kids' day-to-day lives. Because what we learn in the lab and what we learn just being with those kids, it helps these kids grow, and it helps us help the parents to just help their kids every single day. And I'd like to thank Mrs. Latham and Dr. Nutero for letting me into their labs and classrooms. And a big thanks to Dr. Richards, because I almost did not find this mentorship. I got in at the very last minute. And he happened to help me find both of these wonderful ladies, and Mrs. Hyatt for letting us stay during IRSP, and then just the John W. Keyes Speech and Hearing Center, because they saw me a lot this year. So any questions?
Great job, Jenny. I have a question about the sessions over the summer when you were working with the kids in the at the daycare center. Did you, or did anyone like talk to the parents? And I mean, were they able to tell you how the kids did when they weren't at the center? Like, were their behaviors at home changing as a result of the stuff you guys were doing?
Yeah, every day when the parents would drop their kids off or pick them up, we'd let them know what we saw. We'd let them know what they did good or if they were having a hard time. And sometimes if we noticed the kids were having a hard time, we would just ask the parents, like, if something is going on at home. In one instance, actually, we noticed that one of our most energetic kids, he wasn't doing so great. He was always tired. He just wanted to lay there. And we found out from his parents that they think he was actually going through a growth period because he was sleeping a lot at home. So that was really helpful for us because then we knew how to act around him more.
Okay. Dr. Gibbons asks, do you know what statistical techniques have been or will be used to analyze the data from the stutter trial?
For the stutter trial, we actually didn't get very far because one of the hard parts about that was just finding kids who stuttered. I think I only got to look at 3 trials, and one of them we already had to toss out because we had to contact at least 40 public schools within the area and all of their speech pathologists, but the amount of kids that actually reached out wasn't very much, so we didn't get to analyze much of the data at all. We just collected what we could. And I haven't gotten to talk to Dr. Nutero since, so I don't know what we're gonna do moving forward.
Okay. And then let's see, Rohan says, do you have the neurotypical children, do they have similar tests as the children with communication issues?
So the neurotypical child that was in the summer classroom, she wasn't a part of the tests that we did for the kids who stutter, but in the classroom, we'd often have her do the same activities as the atypical kids, and sometimes we'd have her. I do remember one day where we had her take the same test as the on the spectrum kids just to see where she lay. But once we started getting into the everyday learning and activities, because she was a neurotypical child, we had her do slightly more advanced work just to keep her engaged with the rest of the kids.
Okay. And then Dr. Awad asks if there are different levels of stutter, and is it possible to isolate it when kids have other disorders?
I'm not entirely sure if there's different levels of stuttering. There are some kids who can control it better than others. And as for kids who have other disorders, from what I've seen, we are only taking in children who just stutter. If they are atypical or have another neurodegenerative disorder, we won't have them in because the main focus is just children who stutter.
Okay, great. Then if there are no other questions for Jenny, we will move on. Our next presenter is Kunal. Kunal, are you there?
I am here, yes.
All right.
Can everyone see that?
We can.
All right, cool. So hello, everyone. My name is Kunal Patel, and my mentorship took place at the OU Health Science Center.
Kunal, one thing, you're not in full screen mode.
Oh, I'm not?
Yeah. Or we're just seeing the PowerPoint editor window.
Okay, let me see. How about now?
No. Well, it went black for a second and then it went back to the.
Let me try this. Is that it?
No. Oh, wait, there it goes. Yep, that looks good.
Cool. Okay, there we go. All right. So hello, everyone. My name is Kunal Patel again. My mentorship took place at the OU Health Science Center, and I worked under Dr. Jay Hannes. And I would like to give a special thanks to James Hawker and Christian Vannerith for working with me in the lab. My presentation today will be about serum mass profiling for screening and monitoring of cardiovascular disease and epilepsy. One thing that I would like to address first is, before we continue, is what exactly is serum mass profiling? Although this procedure is used globally, many researchers have developed different techniques to undergo this procedure. Serum mass profiling in our lab is the process of analyzing serum of test subjects, which in our case were CABG patients, which stands for coronary artery bypass graft, and epilepsy patients. These two experiments were conducted separately, but I was a part of both during my time in the lab. And so the process by which we analyze this serum is through mass spectrometry. So mass spectrometry is an analytical technique used to identify functional proteins found in a medium. This tool calculates the mass to charge ratio of one or more protein molecules fed through the machine. This mass to charge ratio can be used to separate molecular weight by ions. Mass spectrometers are utilized to determine the exact molecular weight of an unknown compound, which is injected into the machine. So how does the mass spec perform such a calculation, you may ask? Well, it is through a three-step process. Those three steps are an ionization source, mass analyzer, and an ion detection system. Firstly, the protein molecules are ionized or converted into a gas phase ion. These ions produced by the machine, which in our case was called electrospray ionization, will then go through an electric and magnetic field. When the molecules are converted to a positively or negatively charged ion, the machine will accelerate the coupled ions through an electron beam source. Then the ions will deflect through the magnetic field and will then be detected by a mass analyzer. After the detection period, through a computer program, we are given a graph. Each peak that is found on the graph represents a particular ion. These ionization peaks each have a value attached, which corresponds to the mass to charge ratio of the unknown molecule. From there, we can evaluate these values and come up with a list of proteins that are hit by the mass spec. Most of the time, like in this graph, we see a high first peak, which this is typically the buffer that is injected into the machine, which is used to reset the machine each time it's used. The buffer acts as a control variable for ion representation, so for further protein analysis. Now that we know what mass spectrometry is and how to read the graph, we can dive into our lab's research and what we conducted in our lab. Like I said before, we first collected serum samples of our test subjects. Serum is the fluid and solute component of the blood, which does not play a role in clotting. In blood serum, there are carrier molecules which help with the transportation of substances. The reason why serum is used in our lab is because it lacks the protein fibrinogen. This helps us achieve better ionization peaks through the analysis of other proteins. After the blood is centrifuged to retrieve the serum, the substance is extracted and along with its functional proteins are embedded. The proteins attached to the serum antibodies will go through the mass spec for further evaluation. And with the serum samples of multiple CABG and epileptic patients, we found several functional proteins which were hit on multiple patients. So this might be a kind of a large list and you might get overwhelmed by it, but in this list, you see the amount of functional proteins which are found in the serum samples. This is just a sample amount of the proteins. There's about a list of 600 plus in which we dealt with. The symbols that are indicated in the middle of the list, as you can see, IgH or immunoglobulin heavy chain, IgL, immunoglobulin lambda, and IgK, and so on, are the functional proteins detected by the machine. Now, if you look with me on the left side of the chart, we can see the wide-ranging values that spread the chart. The first number in each box indicates the unique peptides which form the functional proteins. The second value, which is enclosed in the parentheses, is the total peptides discovered from CABG patients with type 2 diabetes. And third value is for peptides corresponding with patients not having type 2 diabetes. These values are significant because they help us analyze correlations between the actual hits we get from the mass spec and potential CABG relations. If you look with me now on the right side of the chart, you can see the number of serum samples collected out of 10 that either had type 2 diabetes or did not. This value is especially important for me because it helps me find correlations between the functional protein and cardiovascular disease. Similarly, we have another functional protein list for epileptic patients. However, in this instance, instead of a relationship between cardiovascular disease and type 2 diabetes, we see a correlation between idiopathic epilepsy and neurocystic sarcosis. The goal of this study was to test the extent to which patients with NCC epilepsy of unknown etiology, idiopathic headaches, and among different types of neurocystic sarcosis lesions could be distinguished from each other based on serum mass profiling. For this, we collected sera from patients with neurocystic sarcosis associated epilepsy, epilepsy of unknown etiology, recovered NCC, and idiopathic headaches, then perform binary group comparisons among them using electrospray ionization. In this chart, we only deal with the amount of hits by the mass spec and control hits, which are the unique protein itself. So you might be asking, what am I doing with all these values? So these are where my responsibilities came in. I was tasked with research with BLAST, NCBI, and PubMed research, finding correlations between these functional proteins and cardiovascular disease and epilepsy. I'm not sure if everyone is aware of these databases or not, but if not, these three, BLAST, NCBI, and PubMed, are research databases that have analytics on genes and multiple protein data points. The reason I used these databases was to find the correlations between the functional proteins listed and potential threats to cardiovascular disease and epilepsy. For example, I'll just use IgL. We found that IgL had been found to be abundant in coronary heart disease subjects because of low density lipoproteins and lag phase serum oxidation. This sign given to me can help me find a direct correlation between IgL to cardiovascular disease, especially if IgL is found in lag phase serum oxidation. Another example was T cell receptor beta, where a significant portion of individuals experience a higher risk in peripheral vascular disease with diabetes. Diabetes mellitus is a coronary heart disease, risk equivalent for the outcome of peripheral vascular disease. The impact of diabetes with comorbid risk factors on the outcome of peripheral vascular disease remains unexplored. Like this, I would relate the two factors and conclude from there. My conclusion does not end there because when dealing with 600 plus proteins, there are complicated tracks and paths we need to follow. So there are many proteins found in diabetic patients, and to solve this path analysis is a computer program that I used in my lab, IPA. From an Excel sheet filled with leave one out cross validations and hits from the mass spec, we convert the values to an IPA genetic analysis program. IPA is a web-based bioinformatics application that allows researchers to upload data analysis results from high throughput experiments such as mass spectrometry and any other protein identification tool. In general, lists of genes or chemicals can be analyzed using IPA. It also has a search capability for information on genes, proteins, chemicals, and drugs, and allows interactive building of networks to represent a biological system, as you can see there. In our case, we utilize proteins. So in this chart that you can see, it might seem complicated and discombobulated, but it is actually easier to read and interpret than that list that I showed you earlier. This pathway analysis gives us an easier interpretation model of correlations between a given protein and a debilitating factor or risk, such as diabetes or NCC. From these values, we can create charts, the LOCV charts, or how statistical analysis will generate an independent data set. In these charts corresponding to our epilepsy project, percent total group LOCV classified mass peaks of each subject with calcified NCC, active NCC, or NCC with or without edema or epilepsy of unknown etiology in relation to the cutoff thresholds used to classify a subject into one of two groups. The p-value corresponding to the difference in means of the two groups being compared, and the p-value obtained with the corresponding randomized database. These results suggest NCC epilepsy can be distinguished from epilepsy of unknown etiology based on biomolecular differences in sera detected by our sera mass profile. And due to the ongoing virus, I was unable to really retrieve any more data on our cardiovascular disease project for enough to conclude. So I only have sort of a correlation. And so with that, I would like to thank Dr. Hannes again for letting me work on this project with him and getting to know his lab better. I'd like to thank Dr. James Hawker and Christian Van Earth again for working with me. Finally, I'd like to especially thank Dr. Richards and OSSM for helping me navigate through this mentorship process, providing me the opportunity to participate in a lab-based research mentorship. Is there any questions?
Thank you, Kunal. Great job. So if you did have were able to continue this, what do you think would be the next best experiment to try?
So a lot of my research came with finding correlations between these proteins and obesity, because there's a lot of links between obesity and diabetes. So we found through a lot of different protein samples that obesity was a huge factor in these cardiovascular diseases and why these patients had to undergo a CABG procedure. So I think in the future, to analyze more in that regard, we'd have to kind of dive deeper into the more like the obesity and diabetes relationship.
Okay, great. If no one else has any questions, we will just keep going. So next on the list is Helen and Locke, actually, I think are going to present together. Are you guys there?
Yes.
Yeah.
All right.
Lot, can you share your screen? Okay. Can you hear me?
I can, yes.
Oh, okay. So I'll just start now.
Whenever you're ready.
Okay. Hi, my name is Helen, and my presentation buddy is Locke. We had the same mentorship. Our mentor was Professor Jeff Price at Oklahoma City University, who teaches animation, graphic design, electronic media, etc. Today we're going to be talking a little bit about the software we used during the semester. So I only just started my mentorship this semester, so I'm not going to have as much to say as Locke. I mainly just played around with this 3D modeling software called Maya. I have included a short 5-minute video from lynda.com, which is like the introductory video we all watched to learn Maya.
Let's start off by taking a quick tour of the Maya interface. Now, as you can see, there are a lot of buttons here, but if we break it down into its component parts, you'll find that it's actually fairly easy to navigate Maya. Now, like with any application, you do have a menu bar along the top. We have standard menus such as File, Edit. We also have Create, which allows you to create objects, Select, Modify, Display, which controls how you display objects, as well as Windows, which gives us access to a whole bunch of different types of editors and that sort of thing. And then once we get past Windows, we're going to have a bunch of different menus. In this case, I have Edit Mesh, and this means that I'm in the modeling menu set. Now, Maya has so much functionality that it can't fit everything into this one menu along the top. So we have our second interface item here, which is this pull-down menu, and I can go to any number of different menu sets. So in this case, I'm in modeling, but if I want to, I can go to rigging, and notice how all of these menus on the right side change. If I go into animation, I get all of my animation controls. If I go into rendering, I'll get rendering controls. Now, if we continue on this line here, you'll see we have a number of graphic icons that we can use, and these just mimic what we see in some of the menus here. So we have File Open, we have Save Scene, we have Undo and Redo. We also have some snapping options here that allow us to snap to grid and so on. We also have some rendering controls here. These allow us to render the scene. We have a window here which allows us to sign into Autodesk. And then along here, we have some buttons that will change the right-hand menu. I'm currently have this button open, which is called the channel box. But if I want to, I can click any one of these and get a different set of menus. So we have the channel box here. We have some tool settings. Now this one actually comes up as a floating menu. So if I have different types of tools selected, then this menu will change. I also have what's called the attribute editor, which is very important, and this allows you to manipulate objects a little bit more deeply. For character animators, we have character controls, and we have a modeling toolkit for those who model. Now below this, we have what are called shells. Notice how we have this tabbed interface with a graphic representation of most of the major tools. So if you wanted to model curves or surfaces, you would select this tab. This tab allows you to model and manipulate polygons. We also have an animation tab and a rendering tab. And we can also create our own custom tabs if we want. Now along the left side, we have a number of standard tools here. We have a select tool. We also have a lasso select, which allows us to lasso something, and a paint select. So these allow us to select objects. And once objects are selected, we can move, rotate, and scale. And we'll get into these a little bit. Now we also have down here a number of preset layouts. Now right now I have one big viewport here, and this is what we would call a viewport. This is where we actually work. But as you can see, we have a number of different layouts. So if I wanted to, I could click on this button here, and this will give me a 4-panel layout. If I do this one here, it gives me 2 panels. This one gives me what's called the outliner along with some other panels. And if I want, I can go back to my standard panel. Now these panels can tear off, so I actually brought the outliner up, but I can left click on this and drag, and that will drag that off. And so if I wanted to, I could close it that way. Now along the bottom, we have a time slider for when you do animation, as well as a range slider. So we can actually zoom in on the time slider by left clicking and dragging this bar here, and then we can pan through that as well. Now along the right here on the bottom, we have some playback controls. We have playback speed controls here, which is really nice. And then we also have some additional tools here for looping and so on. Now along the bottom, we have a spot where we can actually type commands into Maya. We won't be getting into that. And then at the very bottom, we have a little helpline, and that will actually just tell us what various tools will do. So if we hover over a tool, notice how these change. So if you don't know what a tool is, just go ahead and keep this little tab open and you will get some hints. So as you can see, my interface looks complex, but if you break it down, it actually is pretty straightforward. And as we move forward, you'll start to learn how to use Maya, and it will become very natural to use.
Okay, sorry, that went on a little too long. So aside from watching the Linda videos, when I was just starting, I found it really helpful to look at step-by-step instructions like this one. I know in general that modeling faces is supposed to be really hard, but I found this tutorial in particular very helpful. Just to get used to how to transform a simple polyhedron like a cube into something complex. You can see how they utilize different tools, etc. In Maya, you can also add colors and render things, but I did not do that because of time constraints. Okay, so these are my own projects. For the cat, I kind of got bored midway, so it ended up looking kind of like a block. For the sword, unfortunately, I am really bad at bookkeeping and I forgot to take pictures of my final work. So I only have these. You can kind of see the progression and how I did things. I first created a long rectangular block and then tapered it at the edges and did the fancy hilt stuff. And then I mirrored it so that the sword is symmetrical. So the rocks, I think, look pretty good, but actually I just pushed and pulled the vertices until they looked right. Honestly, I think the tool that I used the most was the edge loop tool, which lets you insert an edge that follows the geometry of the object. It is very useful for creating more edges and vertices to work with. And also when you smooth your object, it makes it more defined. I also included the picture that I was working from. I think the end result was pretty much like that. So you can use your imagination. Sophie, who's not presenting today, was doing Unity. So I sent my final product to her and she was trying to animate it to make the sword come out of the stone. So pretty cool. So there was one day where everyone in the class went to the OCU mocap lab because they were just starting their motion graphic unit. And they were explaining how all the cameras worked and how the information got onto where it was recorded onto the screen. They had me and one other person put on the really tight black Velcro suits with all the stick-on balls and stuff that reflect on the cameras, and that's how they work. So unfortunately, we only had half an hour, so I pretty much just put it on and then took it off. It was still fun though. I wish I had gotten a picture, but I forgot. So you can just superimpose my face on that person.
Okay, now that Helen has talked about Maya and the mocap lab we did in one day, I'm going to be talking about Unity. So Unity is a cross-platform game engine developed by Unity Technologies back in 2005. Originally, it was only for OS Mac, like the Mac OS originally, but as of now, more than 25 operating systems are able to use Unity. And the engine can be used to create 3-dimensional, 2-dimensional, virtual reality, and augmented reality games, as well as simulations and other experiences. So if you see all those arcade machines with all these cool 3D looking models and all these augmented reality apps on your phone, they are most likely made by Unity. And this engine is also adopted by industries outside of video gaming, such as film, automotive, architecture, engineering, and construction. And I am more interested in the film and engineering aspect of Unity. So while I was in this mentorship, I was trying to learn C# because at OSSM we learned Java, but we didn't learn C#. So I had to pick up a new programming language. And I was about to make like scripts for my program in Unity, and I was about to like rework the Unity engine, but due to time constraints, we aren't able to do that. Another important thing to point out about Unity is we can't really make like character models in Unity. We can only really make an artificial environment for our models and animate models to our liking. We could also add some effects to our landscape, but we can't really like actually like sculpt a model or like a building. So I'm going to be talking about my first model, which is this Pikachu right here. Of course, it doesn't really look like a Pikachu because I'm just starting to learn. But characters such as Pikachu are very difficult to model compared to if you were modeling a building or an object. Characters are difficult to model because you have to take into account of the head, body parts, and then you have to make sure like they're symmetrical so that you could use the model for animations in the future. So this Pikachu actually started off as a cube that eventually expanded itself. I use mostly to insert edge loop, and it's where, like Helen said, where we insert like edges onto a shape so that we turn one cube into two cubes stuck together because we have like this, we inserted an edge somewhere in between like a gap. And I also use an extrusion tool where I take a face, like you see these squares right here. So I took one of those and I extruded them so like I could expand like on the face and it like outwards. So I used that tool multiple times to create the arms and the tiny bitty fingers that are in, or claws in the hand. And I used the mirror tool because I sort of cheated a little where I only like made half of the model and then I started using the mirror tool so I don't have to like do the exact same thing on the other side. And I also used the combine tool because even though this Pikachu started off as a cube, the ears and the tail were not actually part of the cube. I had to make like I have to morph separate shapes and then attach it to the cube using the combine tool. And because characters are difficult to model, this took around 60% of first semester to make. And it also reached out to second semester. And due to the unforeseen circumstances, I cannot texture this model, but I tried my best making the model and it is complete and I really do like the end product. So I also modeled the Dan Little Residence Hall. It's easier to model than most characters, well, probably all characters. But instead of like making this into like starting from one cube, I use a lot of geometric shapes to create this building. So like, for example, like one of the wings right here is one cube, but this base is like another cube. And I at first intended to like model this as like a Hollywood facade where you only see like the front, but you don't see the back. But I eventually changed my plan to that. And I also used the same tools as I did with my Pikachu, the insert edge loop, extrusion, mirror, and combine. And it only took 2 weeks to make this one compared to the Pikachu due to how simple it is comparatively. So Maya and Unity are different programs. I used Maya to model, but I used Unity as my main program second semester. And models, I have to export the model from Maya to Unity by changing the file type. A Maya file type is a .mb file, and I converted it to a .fbx file so that I could bring this model to Unity. And obviously, like compared to the shading right here, the shading in Unity is different because it takes into account a light. And the controls between Maya and Unity are relatively the same. So I didn't have trouble like learning Unity because the controls are almost the same as Maya. And even though, as you can see here, all there is a sunset and it's just a building in a checkerboarded landscape. I made a terrain in Unity rather than in Maya. I found it easier to make a terrain in Unity. So I did that. And so as you can see in the final product, there is like a grass here and then there's a sky. And I used, I put some clouds in. I normally use textured images to help me build the terrain. So here's the quote unquote finished product. So I added windmills as a prop because it was my side project from a while back. So I included windmills as a prop before spring break, before like my last day of mentorship. So in my final product, I added gravity, wind velocity, etc. So like if I were to like activate the simulation, you would see like the windmills like spinning around because of the wind velocity. And unfortunately, due to unforeseen circumstances, I wasn't able to expand the residence hall to its full potential image. I was planning on like building the A wing and the B wing like right next to what I have so far. And that's pretty much how it ended. So I also attached a video of what Unity could do. Yes, it's an anime game that is on a mobile app, but it is used by Unity, and it uses its own like modified engine. So I'm going to be only showing like a couple seconds of it before I end this presentation. So here you go. Okay, so me and Helen would like to thank the OSSM mentorship program, because without the mentorship program, we wouldn't have this unique opportunity to learn these programs. And I would like to thank Dr. Richards for helping us set up this mentorship program. And we would like to thank Professor Jeff Price for taking his time to teach us these programs, and OKCU for letting us use their computer labs. We actually were able to use like a very good desktop computer with 2 monitors, one where we could actually touch the screen. So it's pretty cool using that. And I would also like, I didn't put this on here, but we would like to thank Ms. Falaktu for the assembly program. Without that, we wouldn't be able to be set up with this mentorship. So thank you for listening to our presentation. Are there any questions?
Good job, guys. I have a question. So you said that some of these software programs you can use for like engineering type of projects. Like how would you use this sort of thing in that context?
So I'm actually not too sure about how Unity could be used, but Maya models can be exported to AutoCAD because they are made by the same company, Autodesk, and it also has like simulations where it could test out some real what's the word for it?
Like, will it model the physics and like the stresses on materials and stuff like that?
It does model that. I know because through Maya and Unity, actually, I was able to like set up model gravity to negative 9.8, as we know. And I was also able to like add more factors into the program, such as wind velocity, and I could also incorporate some external like factors, such as I could possibly simulate an earthquake.
Cool. Let's see. Dr. Awad asks, is there any limitation in making a video like the one you showed versus an interactive game with players? Can you do that sort of thing in Unity, or is it just for modeling?
Unity, yes, it could definitely do something like that. And Maya also has the capability to do so. And there are external programs out there that has a capability to do so. I actually learned how to animate a long time ago, like using 3D models using a program called Cinema 4D. But yeah.
Okay. Great. All right, well, if there are no more questions for Helen or Locke, we will move on. It looks like the next presentation will be from Grace Wu. Grace, are you there?
Hello, Dr. Richards.
Hello.
Can you guys see that?
We can.
Okay. Hello, my name is Grace Wu, and for the past 2 semesters, I did my mentorship under the guidance of Mr. Jordan Palmer at a company called Augsburg, and Mr. Jordan is a software engineer there, and today I'll be mainly talking about the application of SQL in determining consumer product utilization, which was my first project that I worked there. So first, I want to talk a little bit about Augsburg and what they do. So Augsburg is a data intelligence company based in Oklahoma City, and they primarily work with energy information, particularly oil and gas. Basically, they go out, they collect information from state, federal, and county records, and then they organize that information and make it more accessible and easier to analyze, among many other things. This kind of information would be valuable to a wide variety of people. Some of their potential clients could be engineers, geologists, analysts, etc. They mainly cover areas in Oklahoma, Texas, and New Mexico, but they're working on expanding that coverage. So they deliver this nicely packaged data through some of their products. 2 examples would be Sol and Atla. So what is SQL? I was actually mispronouncing it until prior to last week when I Googled it. So if I refer to it as SQL, apologies for that. So SQL stands for Structured Query Language. So it's a programming language that's very popular for managing data in a database system. So I spent a couple weeks learning how to use it. So the goal for this project was to be able to visualize trends in consumer product utilization. So using the data that we have in Augsburg's database, we want to organize it and present it in a way that's easy for us to analyze. So this is where SQL comes in. We used it to query a Postgres database, and I'm pretty sure that just means that it's a database that complies with SQL queries. And then we used Zeppelin, which provided the platform to carry out those queries and present it in a nice way. And by nice way, I mean like graphing it. So how this works is whenever a customer would interact with a feature using Augsburg's product, that action would be stored as a new event in the database with like a timestamp and a short description of what that activity was. So an example could be like every time the customer would use like a search option to find like a specific area to look at, among other things. On the left is like an example, like a picture I found for Zeppelin. You can, once you organize like the data, you can choose what categories you want to divide it in to visualize it. So when you organize that, you get something like this. This is when we organized it into a line chart to show the number of events that occurred by document type in Atla over a period of time. So I'm not actually allowed to share what the data is since it is private to Augsburg, so that's why it's all blurred out. But this is what we got when we organized the data by document type, and that represents the different types of actions a customer performed. We looked at the total number of events for each document type category and looked specifically at Atla, which is one of the products developed by Augsburg. And so Atla is a visualization tool that helps map out and filter data relating to oil. I worked on this project back in the fall semester, so we focused on information given for like time prior to 2019. So I didn't actually like see this particular graph until a week ago. And so what's interesting to note is that you can kind of see how the pandemic impacted the usage of Atla. So as you can see, the amount of usage peaked in January and has been declining ever since. So this graph, you can kind of see the data when it's organized by document type and state, those being Oklahoma, Texas, and New Mexico. And when it's like this, it's a bit overwhelming, dividing your data into too many variables. So it's better to narrow down your scope. So this is when we narrowed it down to just Oklahoma. However, this covers document types that are not just limited to ATLA, but could include like their other products. For example, maybe like Sol, like I mentioned earlier. These are the same, but for other states like New Mexico and Texas. So something that you might have observed is that, well, SQL can be used to determine how much a client uses a product over time. It's still up to us to analyze those trends and identify what the causes of those trends are. So you could test this out by maybe like changing an aspect of the product and seeing how the amount of usage is impacted by that. So this could include like maybe changing the design of the tool or adding more capabilities or maybe trying to improve its overall performance. Back when I did this project in the fall, I saw some trends for 2019, but I wasn't actually sure like what the causes of them were. I assumed that when I left for a winter break, the employees at OSSberg were able to utilize this. And I'm really glad to see now how like the report helps show how cause like COVID-19 impacted their product utilization. So before I wrap up my presentation, I want to talk a little bit about the second project I was working on before spring break. So I was working with GIS, which stands for Geographic Information System. It's a computer system, and like the name suggests, it is used to view, analyze, and manipulate geographical data. This is really helpful for editing data in maps and trying to focus on what you want to see in particular. I was learning how to use the application QGIS. It's a GIS tool, and you can install it on like your laptop or desktop. And like I mentioned earlier, it helps manipulate geospatial data. This is a picture I found on Google. It's an example of one of the versions of the application. So basically you could input your own map and add specific components like lines and shapes and many other things. It's really convenient because on the left-hand side, you can manipulate the layers as you please. If you don't want to see bridges, you can have that option. If you want to change the colors, you can. It's like endless options. You can see how the different types of lines represent different things like bridges and streets. And with the random shapes, they can be used to map out lakes or other areas. For OSSberg's purposes, I'm assuming you can use it to map out areas with maybe oil deposits. So I presumed that I was going to be using data from OSSberg to create maps of certain areas in Oklahoma, Texas, or New Mexico to depict these areas with oil deposits. This would be really helpful in deciding like the next place to extract information from and organizing it and preparing it for clients. This could also be helpful for like comparing different areas in those states. So yeah, that's about all I did. So I would like to thank Mr. Jordan Palmer for dedicating his time to mentoring me and overseeing these projects. And I'd also like to thank the other OSSberg employees for helping me understand more about what they do at OSSberg, and also to Dr. Richards for organizing the mentorship and taking time out of his day to drive me to OSSberg every week. And yeah, any questions?
Thank you, Grace. Good job. You mentioned that the data that the software uses comes from like just state and county records, right?
Mm-hmm.
But how does it get in there? I mean, is it do the states just send like a spreadsheet and then you import that into the file, or is it a more manual process where someone has to go through a bunch of documents?
There's actually like, correct me if I'm wrong, my mentor is here, but I'm pretty sure that it like varies. So there is like for a while there it's like a manual way to go out and like physically have to go collect them. But I'm pretty sure there's other resources like you mentioned earlier, like having it more conveniently, like on a spreadsheet or something, and being able to get that information.
Okay.
Yeah.
Great. Anyone else? Questions or comments for Grace? If not, we will go on to our last presentation, which is another group. Elena and Jess, are you guys there?
Yes.
Yeah.
All right. Ready when you are.
Acting weird.
Do you guys have a slideshow?
Yeah, just 1 second. I'm
Okay.
loading it up right now. Okay, 1 second. It's being weird. Ah. Okay. Okay. Here we go. Is it working now?
It is.
Okay. Cool. Of staff at OUHSC. Our mentor was Dr. Wendy Williams, and we worked in the Department of Comparative Medicine. The first project that we'll be focusing on is our project with baboons and making edible nail polish for them. And then the second is on making protective jackets for mice, the research mice there. So I'm just going to start with some background on the research baboons in general. Baboons are used extensively in animal research because of their anatomy, and it's very similar to that of humans. And they're often the last animal to be tested before human testing begins. Because they are so similar, they can be used in a wide range of studies, including those for diseases, contraception, immune system, aging, obesity, stroke, epilepsy, and many more. Specifically at OUHSC, there are quite a few studies happening involving research baboons. One of the more prominent ones is a study concerning a vaccine for a deadly parasite.
We can't quite hear you anymore.
Oh, can you hear me now?
We can, yes.
Okay, sorry, I don't know what happened. So yeah, whenever we first got there, we got to witness that study firsthand, and we got to shave some baboons, which was a fun way to kind of get into our mentorship. And that study is actually moving on to the final stages for human testing because of the work done at OUHSC. So, hold on. Okay, so our baboons, our project, baboons are naturally aggressive animals, and they have a very strict, like, natural hierarchy, and that sometimes causes a lot of violence within the enclosures that they're kept in. And because of this, the baboons often have injuries that require surgery or stitches, which, as many of you know, probably can get very irritated and itchy as they're healing. So the baboons have a tendency to either like pick or
We lost you again there, Jess.
Yeah, I don't know what's
may have a bad microphone.
Is it working again now?
It is now, yes.
Okay. I don't know why it's being weird. It was working earlier. So they're naturally aggressive, and because of that, they often get injuries in their enclosures. And because they can get irritated and itchy, they often pick or bite at these injuries, which can pull out their stitches or make their injuries worse, which is not good for them. So our goal in this project was to try and distract the baboons from their injuries and get them to pick at the nail polish that we provided rather than making their injuries worse. We originally got this idea from Dr. Williams and Dr. Lane in the department whenever we heard about the issue in the summer, and we actually watched them apply regular nail polish to one of the baboons there. So making the nail polish was quite a bit more difficult than we had anticipated at first. We started Googling many different things, and we watched a ton of YouTube videos to try and like lock down a recipe for what we were going to use, because we had to make sure that it was going to be both effective and safe for the baboons. So once we had a number of ingredients that we thought would work, we started trying to find the right amounts that would work for our recipe. There were a lot of issues with our first couple batches because we either couldn't get it to dry fast enough on the nail, or we couldn't get it to stay on the nail long enough. We ended up trying about 20 different recipes before we narrowed it down to 2 that worked best. So these were our final 2 recipes. The major differences between them were the addition of heat during mixing and the quantities of the ingredients that we used. So we had a number of different variables that we were testing in our project. The main one was the differences between the 2 recipes that we used, but we also tried a number of different colors and groups of baboons. So when comparing our data, we focused on the injury status, gender, and age of the baboons that we tested. And because we were not constantly at mentorship, the staff at OUHSC had to apply the nail polish for us and record their observations on a data sheet that we created. So we actually had some pretty significant results from our project. When collecting and recording data, we focused on the number of days that the polish stayed on, and we use that as kind of an indicator as to how well it would work, because not all of the baboons that we tested were injured. So it typically stayed on from either 0 to 3 days, and then there were significant differences between the types of polish that we used, so the 2 recipes that we had, and how long it stayed on between the 2 different age groups that we used.
So this is a graph of all of the different variables we were looking at while we were collecting our data. So we didn't really know what things would make a big difference in our information, so we were just looking at a lot of different variables in the beginning. So you can see that there's a really wide range of how long the polish stayed on average based on different factors. So this is a graph of the difference between how long the polish stayed on the monkeys that were under 15 years old versus over 15 years old. So you can see that the polish stayed on longer on average on the monkeys that were over 15 years old. This is a graph of the difference between how long the polish stayed on our 2 different recipes. So the polish stayed on a lot longer on our recipe number 3 on average than our recipe 15. So our mentor has submitted an abstract of our project, and if it gets accepted, we're on our way to having our information published in the scientific community. So our other project involved the mice used in research. So mice are often used in research because their genes are very similar to human genes. So this makes them very useful for the beginning stages in testing in human studies. And they are specifically good for aging studies, obesity studies, and other things like that. So the mice at the OUHSC are in a wound healing study.
and they have to wear these protective jackets so that they don't scratch at their injuries and interfere with the information they're collecting on how their wounds heal. And these jackets, the ones they have now, are very stiff and pretty uncomfortable for the mice to wear, and they don't move around a lot in them. So our project is redesigning these jackets, and our goal is to make them more comfortable and movable, but still protect the wounds for the study. So our process of making the jackets involved looking up the designs of some existing jackets and brainstorming ideas for different fabrics we wanted to use and different designs we wanted to test out. And then we started making the actual prototypes of the jackets. And once we found a couple that we wanted to try, we started trying them on the mice. So the jackets were actually very hard to fit the mice because we had to figure out a way to be able to get them on the mice, but still have them tight enough to stay on the mouse after it was put on. So we ended up going back and making adjustments and making more prototypes and then trying them on and then going back and making even more. So these are pictures of some of the jackets we tried. The gray jacket on the right is made out of felt, and the black jacket on the left is made out of just a cotton t-shirt material. So one of the things we did was we put an extra patch on the inside of the jacket to over where the injury would be, so to better protect the wound. And this was usually made of just an extra layer of felt, and we also had some waterproof material that we put on top of that. So one of the strategies we ended up using to be able to get the jacket on the mouse while also having it tight enough fitting on the mouse was we threaded the string through the two sides of the jacket, but we kept it very loose so that we could fit it on the mouse. And then once it was on the mouse, we pulled the ends of the strings and tightened it on to fit onto the mouse. So we did not actually have time to complete this project because our mentor short, but our mice, and then in collecting data on which jackets worked best. Dr. Williams and Dr. Lane and Courtney for helping us with our mentorship, and then finally Dr. Richards for organizing the whole mentorship program.
Very nice. So actually, Dr. Williams asks, what were your biggest takeaways from each experiment?
For me, with the baboons, I think it was just really cool to actually be able to try and help the animals in some way. So I think a big takeaway for me was like actually being able to help animals and do research in the same kind of situation, which I thought would be like really cool for a future career. And then with the mice, the big takeaway for me was that there's a lot of trial and error in doing research because we did have to go back and make the jackets so many times. So that was kind of a lesson that I got from it.
Yeah, I also really liked being able to do projects that actually benefited the animals there. And I definitely learned that not everything will go to plan, and there are lots of unexpected things that can happen in your research.
Dr. Awad asks, do you like working with the baboons or the mice more?
Personally, I liked working with the baboons just because they are such interesting animals and you can really like tell their intelligence whenever you're in a situation with them. But the mice were also really fun, and I think we actually got more like one-on-one interaction with the mice, not one-on-one, but like where we were able to really interact and be close with them because the baboons are so aggressive. You have to like keep your distance and be very careful around them. So
Yeah, it was really fun making the jackets for the mice. They were pretty cute, but it was also
You cut out on us, Selena.
their behavior too.
Well, I have a question. I mean, and you kind of addressed this a little bit. I've never worked with baboons, but I'm just guessing they didn't just sit quietly and hold their hand out while you put nail polish on. So how did you actually get them to put it on, or how did it get applied?
So for the non-injured baboons that we did, the nail polish was applied during like physical checkups that they were doing. So they had to be sedated during these, obviously. So they were sedated whenever it was applied. So, and then the injured baboons, it was while they were receiving like surgery or getting stitches for the wound that they had. So they were sedated the whole time.
Yeah. And then I assume, for both the baboons and the mice, were they caged by themselves?
The mice, they're caged with, they have a cage mate, so there's one other mouse in there. There's typically, there can be more than two in a cage, but there's never just one. And then the baboons, they can either be like single housed or they can be in group enclosures. And we worked with baboons that were in both.
So how did they? I mean, I guess in the mice's case, didn't they try to tear off each other's jackets?
Well, we never really got to the stage where they were up and moving around with the jackets because we didn't get to finish the project. So we only had one mouse actually moving around in its jacket because that one did fit so well. But the other times that we tried the jackets on the mice, they were sedated. So.
Okay, cool. Well, are there any more questions or comments for Elena and Jess? If not, that is the end of the symposium. So I just want to thank everyone again for tuning in and for tuning into the end here. I did record the session. I'll make it available online somewhere as soon as I get it processed. So if you'd like to see it, just send me an email and I'll send you a link once it's ready. Again, thanks to all the students for your hard work and for putting together the presentations for today. And as always, thanks again to the mentors for all your time and dedication, to any of those other faculty that are here, those of you who helped me drive and help accommodate the students' schedules so they could do this research. We really appreciate it, and hopefully next year, those of you juniors that are listening in will get to do this in person, but you never know what the future holds. So thanks again, everyone, and if I don't see you again, have a good summer.
Good job, everyone.
Thank you, Dr. Richards.
Little clapping icons.
Great job. Bye-bye, everybody.
Bye, Dr. Lee.
Hey, thank you,
job,
Brent.
everyone.
That was lovely.
Bye.
I don't know how to get the little clapping hands or.
Oh, well, at the bottom, there's a little thing that says reactions. There's only two choices. You can either clap or give a thumbs up.
Okay, I don't have the reactions option. Maybe they blocked me from doing that.
Maybe under the, you may have to have the right view open, but it's okay.
All right. Take
Thank
care.
you for recording the snippets. I'll put once I get the full version processed, I'll post it on YouTube.
No problem. Sorry I arrived late this morning. I forgot.
That's what.
Thank you.
Bye.
Bye-bye.